Ever look at your family and wonder, how come my brother and I are so different, even though we have the same parents? Well, the answer is hiding inside your cells. It's this beautiful, super complex process, and today we are diving deep into it. We're talking about meiosis, the amazing cellular dance that shuffles the genetic deck and makes every single one of us totally, completely unique.
So here's how we're going to break it down. We'll kick things off with the basics, one parent or two. Then we'll get into why meiosis is the absolute key to sexual reproduction. We're going to follow the chromosomes through every single step of their dance, see how that dance creates all this amazing variety and, what happens when things go wrong. And finally, we'll zoom out and see how the rules of this dance are crucial for our health in general.
All right, first things first. Let's look at the two main ways life makes more of itself. It really boils down to a simple choice. Do you make an exact copy, or do you create something brand new? Okay, so this table lays it all out pretty clearly. Asexual reproduction, think of it like a photocopier. One parent, and it passes down all of its genes to make a perfect clone. But sexual reproduction, that's a collaboration. You've got two parents, and they each contribute only half of their genes. This is what gives you a unique individual, but it also creates a huge math problem. I mean, if you just smash two whole cells together, the chromosome number would double every single generation. So how does life solve that?
And that brings us to nature's seriously elegant solution. The process that solves this exact problem is called meiosis. It's what I like to call the great reduction, and it's what makes this whole two-parent thing possible.
So what is meiosis, really? It's a special kind of cell division, and it has two main jobs. Job number one, cut the number of chromosomes in half. Simple enough. But job number two is where the magic happens. It makes sure that the four new cells it creates, what we call gametes, like sperm and eggs, are all genetically different from each other. And here you can see that beautiful cycle in action. Look, an adult organism like us is diploid, or 2N. That just means we have two sets of chromosomes. Meiosis is the process that creates these special haploid cells, the sperm and egg, with only one set, or N. Then, during fertilization, they fuse together and bam! The 2N number is back, right there in the brand new zygote. It's this perfect system of having and then recombining. It makes sure every generation starts with the exact right amount of genetic material.
Okay, so we know what meiosis does. Now let's pull back the curtain and see how this incredible dance actually happens inside our cells. It's a two-part performance, meiosis 1 and meiosis 2.
Meiosis I is all about the pairs. First, the homologous chromosomes. That's the one you got from your mom and the matching one you got from your dad. They find each other. They pair up. And right here, they do a little information swap. We'll come back to that. It's super important. Then, the cell pulls those pairs apart, sending one whole chromosome from each pair into two new cells. And that's it. That's the great reduction. We've gone from one diploid cell to two haploid ones.
Now for Act 2, meiosis 2. At first glance, this part looks a lot like your standard cell division, mitosis. We take those two cells we just made and we divide them again. But this time, the goal isn't to separate pairs. It's to separate the sister chromatids, those two identical copies of a chromosome that are stuck together. Like splitting the two sides of the letter X right down the middle.
So let's just recap the results real quick. At the end of meiosis 1, you get two haploid cells, but each chromosome is still in that X shape. It's still duplicated. Then meiosis 2 comes along, separates those duplicates, and leaves you with a final product, four haploid cells, each with single, skinny, unduplicated chromosomes.
Okay, now for what I think is the coolest part of this whole story. Meiosis isn't just about counting chromosomes. Oh no, it's an absolute masterclass in creating genetic variety. This is where things get really interesting. Which brings us right back to where we started. If you and your siblings get your genes from the same two people, why aren't you all identical? The secret is a process that shuffles the genes before they even get passed on. And that secret is called crossing over. It is amazing. Back in meiosis I, when those homologous chromosomes are all paired up, they get real cozy. They literally wrap around each other and swap pieces of their DNA. A chunk of the chromosome from your mom physically breaks off and attaches to the one from your dad, and vice versa. Now, this doesn't create totally new genes, but what it does do is create brand new combinations of the genes you already inherited.
And here's a look at how that's even possible. The homologous pairs line up with incredible precision, forming this four-part structure we call a tetrad. You can see it right here. This alignment has to be perfect. It's like lining up two zippers before you can trade teeth between them. This is the setup.
the moment right before the genetic shuffling begins.
And here's the swap. Sections break off and reattach to their partner chromosome. The result is what scientists call recombinant chromosomes. They're not mom's chromosome anymore, and they're not dad's. They are completely unique mosaics, carrying combinations of traits that literally did not exist before in your parents. And since this can happen all along the chromosome, the number of new combinations is just mind-boggling. This is the engine of diversity. It ensures that every single sperm or egg cell is a one-of-a-kind genetic lottery ticket.
But, with billions of these divisions happening, sometimes this super precise process makes a mistake. It's bound to happen. So what are the consequences when this intricate cellular dance stumbles?
Well, the most common type of error has a big name, non-disjunction. But all it really means is that the chromosomes fail to separate like they're supposed to. This little hiccup can happen during meiosis 1 if the pairs don't separate, or it can happen in meiosis 2 if those sister chromatids don't split apart. And when the mistake happens really matters. As you can see here, if that failure to separate happens in the first meiotic division, then every single one of the 4 final gametes will have the wrong number of chromosomes. Every one. But if the mistake happens in the second division, you get a different result. Half of the gametes will be abnormal, and half will be totally fine. Either way, you end up with a sperm or an egg that has one too many or one too few chromosomes, and that's the starting point for many genetic conditions.
These errors in meiosis really shine a spotlight on a much, much bigger idea. Just how incredibly important it is for our bodies to control when and how our cells divide. And this isn't just about making babies. This is about keeping our own bodies running, day in and day out.
So, while a mistake in meiosis can affect the next generation, a mistake in the control of regular cell division, that's mitosis, in our own bodies can cause problems right now. Because when a cell forgets how to stop dividing, when it just starts multiplying over and over and over, well, that's the very definition of cancer.
So, how in the world do you fight a disease that's defined by out-of-control growth? Well, the main strategy behind a lot of cancer treatments is actually to turn that very characteristic against the cancer cells themselves.
Modern cancer treatments basically attack this runaway division from all sides. Chemotherapy uses drugs that poison any cell in the body that's dividing quickly. Radiation blasts a specific tumor with energy, shredding its DNA so it can't copy itself anymore. And of course, surgery is the most direct approach. You just cut the whole thing out. There are 3 different methods, but they all have the same target: that cell's broken off switch.
All right, let's wrap this all up. We saw that meiosis is the crucial step that halves chromosome numbers, making sexual reproduction possible. We uncovered its secret weapon, crossing over, which is this amazing engine for shuffling genes and creating diversity. We also learned that mistakes in this process, like non-disjunction, can have really serious consequences. And finally, we connected it all back to our own health, seeing how the rules that govern meiosis are the same rules that, when broken, can lead to cancer. It really shows that controlling cell division is just fundamental to life.
Which leaves us with one last thing to think about. We've seen just how complicated meiosis is. The pairing, the swapping, the 2 perfectly timed divisions. So what's the real miracle here? Is it the incredible, beautiful diversity this process creates in every new generation? Or is it the fact that a system this complex, this prone to error, somehow manages to get it right almost every single time?